everyone. This is Jyotsna Hegde with NRI Pulse. Today, we are here with Dr. Rafi Ahmed, who is the director of Emory Vaccine Center at Emory University School of Medicine in Atlanta and a Georgia Research Alliance eminent scholar. His research efforts are directed towards understanding the mechanisms of immunological memory and using this knowledge to develop new and more effective vaccines, and also to define the mechanisms of T-cell exhaustion and developing effective immunotherapy for treatment of cancer and other chronic infections, viral infections. Dr. Ahmed is a member of the National Academy of Sciences, the National Academy of Medicine, the National Academy of Inventors, and the American Academy of Arts and Sciences. Dr. Ahmed, it's truly a pleasure and honor to have you with us today. Welcome.
Thank you, Jyotsna. It's indeed my pleasure.
So, Dr. Ahmed, since the pandemic began, we sort of back 2 years ago, we initially heard, the adjective unprecedented go along with it. And then, as time went by, we had a vaccine, but then the virus, as it often does, mutated and people had to get boosters. And the trend sort of still continues to this day. But, here actually to talk about this big breakthrough output, so to speak, with, the Emory Vaccine Center here in Atlanta, along with the Emory Vaccine Program at the International Center of Genetic Engineering and Biotechnology, which is a collaborative program in India, which resulted in this key output. So basically, the output happens to be the coronavirus busting monoclonal antibody, which is effective, against multiple variants. So please tell us more about this.
Yeah, sure. Yeah, so this project actually started in the middle of 2020, when really the first wave of patients were getting infected in India. And so we were contacted by the director of ICMR, who contacted me, and he was aware of our group at ICGEB, our lab over there. I should tell you a little bit about our lab over there.
Sure.
So this was a relationship that was established between Emory University School of Medicine and the International Center for Genetic Engineering and Biotechnology. This is a premier biomedical research institute based in Delhi. It has also an international origin with a connection to Trieste in Italy, but it's fully funded and supported by the Indian government, by the Department of Biotechnology, which is part of the Ministry of Science. Ministry of Science. So when I say we, I mean Emory University and ICGEB plus DBT and the Ministry of Science. We established a collaboration between the two institutions over 15 years back. And the agreement was that ICGEB would provide us with the lab space at their research institute, and would also allow us access to all the infrastructure that they have. This is a really state-of-the-art biomedical research institute, and that that Emory University would invest by having on site people who are supported by Emory University, and so we have a kind of relationship where one of our faculty members spends about 50% of his time there. His name is Murali Kaja Krishna. And another faculty member who's kind of a joint faculty between ICGEB and Emory. Actually, she's based there full-time. Her name is Anmol Chandali. So these are the two people who really get the credit for doing this, Murali Kaja Krishna and Anmol Chandali. Anmol Chandali actually is there full-time. And we have gotten good support for our research from the NIH in the US, and also from the DBT and ICMR in India. So it's been a very successful partnership between an American university and an Indian research institute, with all the research being done on site. Our rationale for setting this up was that one can always have collaborations between universities in India and in the US. And those are also very successful, people going back and forth, exchange programs and collaborations. But we felt that we would be much more successful and if we actually had a footprint right there, that is, we were doing research over there. One of the biggest advantages of this is that you don't have to ship samples, which is always a big problem. So this way, no samples are shipped. We get the samples there from infected people or vaccinated people to analyze immune responses. So this has been a very successful program. It's not a very large group. It's only about 15 to 20 people, these 2 faculty members. And then we are lucky to get very good graduate students from ICGEB into our lab, and also postdocs and research technicians. A small group, but works well together and has been very productive over the last 15 years. So we were actually focusing mostly on dengue virus infection. But as you know, dengue is a huge problem, unfortunately, in India.
That I think the chikungunya and all of these things.
Yes, exactly. That's right. We were also working on chikungunya, and as part of the analysis of dengue infections in India and also chikungunya infection in India, we were doing a detailed analysis of their immune response, their T and B cell responses. And also we had developed their technology that actually originated at the vaccine center in Atlanta of how to make monoclonal antibodies from the B cells actually of the infected patients, or the vaccinated patients. So these are fully human monoclonals that come actually from the patient of interest. And then these can be used for diagnostic purposes or therapeutic purposes and so on. There's a lot of uses having these antibodies for therapy or for diagnosis. So our research program was focused really on dengue for the last 10 years, and we have published and made significant contributions there. But of course, when COVID happened, everything shut down, and that was the period of the severe lockdown in India. You remember, this is the March, around February, March, April of 2020. Yes. At that time, no vaccine was available. So the ICMR director contacted us and asked us if we could try and make human monoclonal antibodies against SARS-CoV-2 from the patients who were infected there. So ICMR provided funding, and of course DBT provided also additional support. So our group got samples from about 20 SARS-CoV-2 infected people in India. It came from different parts of India, and ICMR arranged for the shipment of blood to our lab in Delhi. And then our group, I mean, really, I'm so amazed at what they did, because this was the time of the lockdown. You could barely go out. They had to have special permits with them so they could show it to the police who would stop them. Wow. And then they came, really a heroic effort. And then they came and worked, at the lab. Pretty much all of our group was working there, full time. And so we were able to, or I should say, they were able to make about 300 monoclonals. Monoclonal is a single antibody.
I was going to ask you, in layman terms, what is a monoclonal?
Monoclonal, so I think you can forget about monoclonal. You can just think of it as antibody.
Antibody.
The monoclonal is a technical term that to most intelligent people who are not scientists, it really is a confusing term. Let's say antibody. So we generated a large panel of antibodies, over 300 different, so let's say different antibodies, okay? 300 different antibodies that came from different patients. And then those were tested for their reactivity, for their ability to neutralize the different SARS-CoV-2 variants. Now remember, these patients were infected with the original ancestral strain, that is the Wuhan strain. So the infection is the Wuhan strain, the original strain that emerged. And even the Delta had not emerged. The Delta emerged in June. So this is pre-Delta. Okay. At that time, no new variants had come up. This really was so most of the antibodies that were generated by our lab in Delhi were able to neutralize the Wuhan strain, but did not react very effectively with the other strains. Not surprisingly, some reacted with some, but again, they were the typical antibodies that one gets, which are very effective against the strain that infected them, but are not that effective against the new variants, especially the Omicron variant, and not at all. But we got very lucky. I think this is really a bit of serendipity and luck that that actually two of the monoclonals that out of these 300 or so that were made were very broadly cross-reactive. And what that term broadly cross-reactive means is that they not only neutralize the Wuhan strain, but also neutralize the Delta, the Beta, Omicron, and so on. And one antibody in particular, the one that we published a paper on recently in Science Advances, this antibody is really quite unusual and one of the very rare antibodies that have been described anywhere. And this one not only neutralizes, of course, the original Wuhan strain, it neutralizes the Delta strain, the Beta strain, which is not that surprising. But what is really surprising was it neutralized all the Omicron strains, the BA1, the BA2, the BA4, and the BA5.
Wow.
And not only did it neutralize them, it neutralized them with equal efficiency. So let's say if the ability, if you set a bar and say, okay, that it was, if it was 10 against Wuhan, it was almost 10 even against
Omicron.
the Omicron.
Wow.
This is very rare because most other antibodies, if they neutralize Omicron with value of 10, the one for Omicron would be 1 or even less than 1.
Right.
It would be 10-fold lower, and sometimes 50-fold lower, sometimes no recognition at all. Okay? And we had many of those antibodies too. Okay, like most of our antibodies, but the remaining 290 antibodies were that way. Okay? But then this one, really, we were very lucky, as I said. Hard work by our group, but actually getting this was really quite fortunate. And then a group at Emory University, a structural group at Emory University, Anamika Patel and Eric Oatman, who are structural biologists, they helped in the actual structure of the antibody. And what they did was to find out why is this antibody so effective, and why is it utilizing all these different variants? So it turns out that this is seeing a very unique part of the spike protein. You know that the way the virus is neutralized, it has the spike protein sticking out.
Yeah. We saw that picture everywhere. Yeah.
Exactly. You've seen it in every
Yes.
magazine, every newspaper, every TV show. You've seen it 100 times. Okay? So it's that big thing that's sticking out. Okay? And that's the protein that binds to the cellular receptor. So if that is blocked, then the binding to the receptor is blocked. That is, it can't infect the cell. But this is now seeing a site on that spike protein, which is shared between all of the coronavirus, all of the different variants. And it's seeing it from very different angles. Okay. So most antibodies will bind to a part of the spike protein that is present on the Delta and the Wuhan and the Beta to some extent, but not on the Omicron. This is seeing a site that's common to all of the different variants. It's actually a better monoclonal in terms of its effectiveness than any of the currently licensed monoclonals. So the ones that are being used in the US and other parts of the world are not as effective in terms of their ability to neutralize the different variants. Maybe there might just be an occasional antibody here and there. So what we are now hoping is that the Indian government will work with the appropriate pharmaceutical companies in India. And the way we did the patenting of this antibody, really it's patented in India so that it could be used in India and produced by a pharmaceutical company in India so that there would be cost issues would be taken care of, that it would not be too expensive to buy. Because if you make something and it's too expensive, then of course the people in India don't benefit from it. So that's why we want this to be an antibody that's produced within India by the people who, there is the pharmaceutical company there and then sold at a cost that's affordable. So that's where we are in at this stage. And basically it's the Indian government, i.e. the DBT and the ICMR, with ICMR taking the lead in doing the negotiation.
Wow.
Actually both, I should say both ICMR and DBT jointly working together to develop this as a commercial plan.
So given, this broad range of effect that it has, Dr. Ahmed, so is this antibody specific to COVID, or would the impact
No,
go beyond?
no, it won't go beyond COVID, but what we don't know now is
Mhm.
whether how effective it would be against the next variant or the next variant that
The
we
next variant. Okay.
yes, so we can't predict the future. Okay, so
Right.
we don't,
You
but
don't
give
know.
but given the highly conserved region
Mhm.
that this antibody is seen, we our expectation is that it would also be of some value even when new variants are there. Yeah, but still BA5 and still a very dominant infection at the moment.
Yeah.
Some other variants are coming up, but still BA5 is the dominant one. So I think that if it's produced quickly and is available, it would be of great value to people as a therapeutic and prophylactic, but in particular to immunocompromised patients.
Yeah. Yeah. That's what I was
Patients,
going to ask you. What is its, relevance,
yeah, of
does
course,
it
the
study
vaccine is
neutralizes?
always, yeah, always the vaccine is always better because it can be given to many people
Mhm.
easily. The administration of this requires, a little bit more involved steps, going to the clinic and then getting the injection, the infusion. So this is not for everyone. It's not to be given to everyone, but would be of great value for people who are immunocompromised or or somebody who's actually quite sick and needs the therapeutic treatment. But very useful even as a prophylactic to cancer patients who have cancers of the immune system, people with lymphoma, people with multiple myeloma. They don't respond well to the vaccine. So currently in the US, people with lymphoma or multiple myeloma are also transplant patients.
Right.
Transplant patients who are on immunosuppressive regimens.
Mhm.
In these individuals, even the best vaccines don't work because their immune system is compromised.
True.
So currently, standard of treatment here for people who have these malignancies or cancers of the immune system, actually it is giving them the monoclonal antibodies because that's the way for protecting them. So this is a routine administration of the antibodies which are currently licensed here for transplant patients and cancer patients who have really deficiencies of the immune system. So I think that this antibody will be of great value to people like this in India and also globally. I would yes.
So what is the potential for, commercialization of this as a solution to the public against the pandemic?
It would be, as I said, the main value would be for people who are very sick and hospitalized. This would be very effective, or could be very effective. It would be very useful for specialized populations that I mentioned to you.
Right.
Yes, a antibody therapeutic or antibody prophylaxis is not like a vaccine. You can't take this and give it to everyone because
Yeah.
one of the um, um, I wouldn't use the word disadvantages, but one of the realities of a antibody treatment is that the antibody has a fairly short half-life. That is, within a few weeks, antibody will disappear
Yes.
because there is no cell there that's making it. So if I inject myself or if I inject you with this antibody, we will have sufficient levels of this antibody for about 2 to 3 months. Okay, but then it will start declining. So that's why for these cancer patients and transplant patients who are on immunosuppressive drugs, they get this infusion every 3 months. So that's why it's not a practical solution for like a vaccine. You can't
Right.
be giving this monoclonal antibody, therapeutic antibody, a prophylactic antibody repeatedly to people. It's too expensive, too costly. That's not the. But in the setting of a severe conditions, like for example, if there was no vaccine available, this would be. Fortunately, we do have vaccines which are certainly very effective in preventing severe death, even if they don't prevent infection. So I think it's a so we deal with the pandemic by vaccines in a more broader way, And
Yeah.
then with these antibodies like this for specialized populations and specialized settings where something like this can quickly happen.
So, Dr. Ahmed, where do you see us, in the course of this pandemic, in terms of current variants, infections, reinfections, and boosters?
I think we're at an interesting place right now where I think the real threat of the pandemic is much less now. It's not like it was in 2020, especially where it was a horrible situation because we did not have vaccines, we did not have antivirals, we didn't have any of these monoclonal antibodies for treatment. It was a very difficult time. But fortunately now we have effective vaccines that are, I should emphasize the vaccines are not that effective in preventing infection and mild symptoms, but they are very effective against preventing severe disease. So I think our best defense or the best way of continuing to control the pandemic is still vaccination. And I'm optimistic. I think that there will always be a few people infected and variants will continue to emerge. We will have to learn to live with it like we're living with influenza. So it's a serious disease. Yeah, it's a serious disease. Flu kills a lot of people, especially the elderly. So I think we have to now live with one additional, serious respiratory infection. So this would be in the same category or maybe even more dangerous than influenza virus. But it's not the pandemic situation. Okay. So that's why I think we should be resuming normal life. Okay, but just be careful and make sure that we are vaccinated and are protected, and just follow public health measures. But we're not going to be in a situation where no one gets COVID, okay, and no one is seriously ill from COVID. And unfortunately, people who are elderly, have other comorbidities, or have this I keep coming back to the immunodeficiency, but I think this is where our monoclonal antibody that we made has a real place. And there's a very large number of people worldwide and also in India who are immunocompromised and are either due to these cancers or due to taking immunosuppressive drugs. Look at all the transplantations. All the transplantations there's a very large number of transplantations in India and worldwide who have to be taking immunosuppressive drugs. For them, the vaccines will work. In some cases, will not work. In some cases, because the immune system is being suppressed. And what's being suppressed there is the ability of our immune system to make an immune response. But if we already provide the product, so the antibody is the product of our immune response. So here, we are giving the product. So it doesn't matter if they're an immunosuppressive drug, because the antibody is what's going to neutralize the virus already there. So those people can't make that antibody, but if you give it to them, they will be fine. Yeah.
So how was this, collaboration, between the faculty here, at the Emory Vaccine Center, and its joint India program, critical for this important output?
Yeah, I kind of told you a little bit about this in the beginning to give you the background. I'll kind of repeat that. So this initiative started in the around mid 2000. I should remember the exact date, but I don't.
You're working too hard.
Yeah. I should remember these dates, but it's over 15 years. It's about 17, 18 years. And this started because I've been on this since 1994. This date I remember. Since 1994, I've been on an advisory committee for the Department of Biotechnology in India, just as part of the Ministry of Science. So they had set up a committee of expatriates, that is scientists who are of Indian origin, but are working overseas. And the name of this committee was Scientific Advisory Committee with the O, the letter O in parentheses, and it was basically overseas. It was the Scientific Advisory Committee overseas. And it was a fantastic committee. It still functions, but not in the organized way it was doing at that time. But this.
So that was the origins of this?
Yes. So we used to refer to this as SAC-O. O, because it was the acronym was SAC/O. So the SAC-O was a very outstanding committee because it had about 10 people. It was not very large, and about 10 scientists. 10 scientists in different areas of biomedical research. It was for biomedical research. It wasn't chemistry or engineering or mathematics. It was biomedical sciences. And we had people who had expertise in immunology. So I was the person who was representing the immunology expertise on that thing and vaccines. There were people who were experts in cancer. There were people who were experts in genetics. There were people experts in plant biology. So really a terrific group of people. And about half of them, or about, I'd say only 2/3 of them were from the US. And then the remaining 1/3 were from England, Europe, and other parts of the world. But
I was going to ask you if it was just US. You already...
No. We had people, actually very active participation of Indian origin scientists from the UK. So it was actually, I would say, US and UK were the two main, but then we had people who were from also from Europe, who were Indian origin. And we also had a few people who were in other parts of Asia, in Southeast Asia, Japan, or, Indonesia. Or basically, it was really, truly a global thing. But the common theme was that we were all of Indian origin, we were born and had grown up in India.
Wow.
And we're now scientists in other parts of the world. And the history for this, and this is anecdotal, I of course was not there when this conversation happened, but the history for the origin of this actually apparently goes back to Indira Gandhi. When Indira Gandhi was the prime minister, she asked, there was a cabinet meeting, and she asked the Minister of Science and Minister of Health there, that India has so many wonderful scientists abroad, and they've become very successful. What do they do for us? And the answer was, they don't do anything for us.
So she said, put them to work, huh?
She said, well, why can't we get
utilize
them to help us? Utilize them. So that was the origin. So this would have happened, way before I actually joined it. So I think this SACO started maybe like in sometime in the 1980s, 1970s, 80s.
Very interesting.
Yeah, and then I joined this group in 94. And so
you said that,
yeah. So we would go there, all 8 or 10 of us would go there, and we very much look forward to it. We'd go there, have meet with the leadership there. We were reporting directly to the person who was head of the Department of Biotechnology. And then that person, the secretary of the DBT, would report to the Minister of Science. So we would basically, the whole proceeding would be organized by the DBT secretary, the head of that. We would meet with all the leadership, the leading scientists from the DBT institutes in India would come. Would be a 2-day meeting. We would meet with the Minister of Science. And it really, there was a lot of support, and we would give recommendations. But then also so we would meet once a year, but also throughout the year, we would be in touch with them. Someone like me actually went there at least 2 or 3 times a year, not just for the annual meeting, but at least multiple times, related to some review or some discussion. And it was really through this contact that I started the discussion with the DBT secretary at that time. Her name was Manju Sharma, about this possibility of a relationship between Emory University and a biomedical research institute there. And Veer Chauhan was the director of ICGEB at that time. And so basically,
let me interrupt and ask you why the specific, engagement in India. What, what sort of pushed you to?
Well, we are from India, so we would like to, like as Indira Gandhi said, what do they do for us? And so was very little. Okay, so I think it was really to do something from the country that we're from, and of course there are very talented and very intelligent young men and women there. So it's a place where the talent is there. And of course, institutions are there also, which are great. But we thought that we could also partner with them. And we wanted to partner with them in particular in the area of human immunology, because this was an area that was not very widely being studied here. So our goal was twofold. One was to do it in India because we are from India originally, but also to develop a area of expertise which was kind of underdeveloped here. So although there was a lot of good studies being done on infectious diseases there, a lot of good studies being done on immunology also, but there was really very few people in India at that time. This is again in the late 1990s when we started this, who were doing human immunology. So my proposal to the DBT secretary and there at that time, and then Raj Bhan became the DBT secretary, an extraordinarily visionary individual. And I think that it really was the combination of people like V. Chauhan, who was the director of ICGEB, Raj Bhan, and then the Minister of Science at that time was Kapil Sabil, who was very, very supportive of this. So really, we had support at a very high level for setting up this partnership. And as I say, it has persevered for almost 20 years and has been making contributions. So it's an example of something small, but I think with some vision and filling a gap over there. The reason I got that call from the ICMR director was because they knew that we had the expertise in doing human immunology and making these antibodies from patients' B cells. And that this technology was operative there because we had done the same thing for dengue. We had made a large number of dengue molecules, antibodies against dengue virus. So I think it's been something we're very happy with, and we hope it will continue, and that many other people will duplicate and do something similar.
So what are your future goals for the India program that you have going now?
India program, we will continue with our partnership with ICGEB. We just actually renewed it for 5 more years. We'll continue working on COVID because as we just talked about, the threat is not gone. And we will also continue, and now that we can look back again at dengue, so we will be looking at dengue in more detail. Several dengue vaccines have been produced, and they're not licensed yet for use in India, but clinical trials are going to be done in India, looking at dengue vaccines that have been developed, that are being developed actually in India by Indian pharmaceutical companies. So we'll play a role in analyzing immune responses in those vaccinated individuals. Essentially, our goal is to help in analysis of human immune responses for vaccines or for infections, and developing reagents like this, which can be of value. So we will continue this. We are also trying to extend our partnerships in India with other institutions, and we are very interested in particular in going into the cancer space, in cancer immunotherapy. So our next major step in India would be in the area of cancer immunotherapy. So our vaccine center and people here at, we have a very good cancer center at Emory. Suresh Ramalingam is the director of that. And so Ram and I actually are collaborating with the Tata Memorial Cancer Center in Mumbai. So basically it will continue the infectious disease work at ICGEB, but we also want to really have a major effort now on cancer immunotherapy.
Makes sense. How can the Indian American community what can they do to strengthen these relationships, Dr. Ahmed?
Well, they can support us.
How can they support you? How can they support you?
Well, I hate to do fundraising on your program, but I think that
no, but it's important,
Yeah, it is. I think that so far we have funded this program. Emory has been very generous. So Emory, I should thank the Emory leadership, starting from the president down to the deans and all of that. They have been very supportive and provided funds to the Emory Vaccine Center to support these activities. So Emory's commitment to doing research in India is really very, very impressive, and with full commitment in that. But the actual research has been done by funding either from the NIH or from the Ministry of Science and Ministry of Health in India. But what we have not had so far is a very wonderful philanthropist who will say, you're doing great work, or many wonderful philanthropists who say, you're doing great work, we would like to support you.
Sure, sure. Dr. Ahmed, you're a world-renowned expert in immunology, your journey from India to, the current world stage. What are your perspectives on the future of biomedical science research in addressing future pandemics and, diseases of maybe global concern?
I think it's very good. I think I'm very optimistic because there is huge interest in this area. So a lot of very smart young people are going into this field, and there is actually very good support from government. I think both in the U.S., globally, I would say, in Europe, and also in India too. There's a lot of support for pandemic preparedness, for area of infectious diseases, and just biomedical research in general. And then the third thing, which I think in some ways is the most important in terms of producing product, is that the pharmaceutical industry is investing very heavily. And so they have, and not only just investment in producing the products, but also in research. So the pharmaceutical company has really expanded and improved their research capabilities. New technologies are developing. We saw the development of the mRNA technology by Pfizer and by Moderna. I would say BioNTech and Pfizer, but many others. Many new different cancer immunotherapy approaches are being developed by Merck and BMS and Genentech and Roche. I think as far as biomedical research goes, I think this is one of the most exciting things. I would encourage all young people to go into this area.
Of course, they should follow your footsteps.
They can not only do good science, but also see their science having an impact in the,
absolutely,
in terms of public health. Yeah, I think it's really a wonderful time for this.
Thank you, Dr. Ahmed. Thank you so much for talking to us today.
Thank you. My pleasure.
Yeah, we were right on time. We just have 1 minute left. So thank you so much.
Okay, bye-bye. Thank you.
Bye.